Hello everyone, welcome back in PTK Pathsala. Today we are going to learn about the restriction enzymes. So when we talk about the restriction enzymes, so these are the particular DNA nucleases which play a crucial role in molecular biology. Basically, they recognize a specific recognition sequence, we can say that, or restriction sequence on DNA. And these sequences are palindromic sequence to cut the DNA into small fragments. And after cutting at specific sites, they're going to produce a fragment with sticky or blunt ends. Now we will talk about the discovery of these enzymes. So restriction enzymes were first discovered in late 1960 by scientists when they were studying about bacterial defense mechanisms against viral DNA. So here I will explain you what actually happens is that when a bacterial cell get infected by any viral DNA, then for its own protections, the bacterial cell is going to secrete these restriction enzymes which cut the viral DNA into a small fragments. Now, here the question arises that since these types of like enzymes cut DNA because they are particular for cutting a DNA, so then there are high chances that they will also cut the bacterial genomes. What let me telling you that this does not happens in bacterial cells, we can say that, because to face this problem bacterial cells follow another mechanisms by modifying this their restriction sites, and these modifications occur in presence of methyltransferase, which add methyl group to the particular this here recognition sites, due to which the restriction enzymes are unable to recognize this specific sequence to cut the DNA. Because now after binding, after add methyl group here on restriction sites, the restriction enzymes are unable to recognize the restriction sites to cut the DNA. So this is how the bacterial genome is saved from being cut by these types of enzymes. Now, let's talk about the recognition side. So, each restriction enzymes recognize a specific short DNA sequence, typically 4 to 8 base pair long, and these sequences are often palindromic, means they read via the same forward and backward directions, and the recognition sites of each enzymes is different. And after cutting the recognition sites, these enzymes cut the DNA, and this cut can happen at any sites of DNA strands. Either it may be on 5' or 3' ends of DNA strands. Depending on the enzymes, the cutting ends of DNA can be either sticky or blunt ends. And one particular feature sticky ends have single stranded overhang that can base pair with complementary sequence, while blunt ends are straight cuts. It's not have any types of overhangs. So this is all about restriction enzymes from my sides. See you in the next video. So till then, bye bye and take care.